action of the enzyme hyaluronidase released from the acrosome of the sperm. So why do you have to know it's hyaluronidase specifically? Well, this is an enzyme that catalyzes the hydrolysis or breakdown of hyaluronic acid. And this is important because hyaluronic acid is kind of like the cement holding the follicular cells together. So if you think of the corona radiata like a protective wall or fortress around the oocyte, and then the hyaluronidase is like a tool to peel off the cement holding those bricks together. Which leads to my next point. Weakening the cement won't necessarily cause the wall to tumble. So if you were an invading army, excuse my pun, you would still need to apply pressure on that wall to knock it down, apply pressure on that weak spot after you remove that cement. So how does the sperm do this? Well, the movement of the tail of the sperm, which confers motility to the sperm, isn't just important in getting the sperm to the egg, it also plays an important role during the penetration of the corona radiata. To summarize, for this first phase, in order to pass through the corona radiata, the sperm therefore needs to be capacitated before it ever gets there, and this is the removal of the glycoprotein coat that allows the functional enzymes to be released. Okay? The second thing is you have to have the functional enzymes, in particular, in this case, hyaluronidase, available. But the third thing that's also important is the sperm needs to be motile. Now, the second phase is the penetration of the zona pellucida. Now, imagine you're an exhausted sperm. You have just peeled away the cement, knocked away enough space in the fortress wall to get through. Now you are in this thick gunk of gel, of proteinoid goo, if you will, called the zona pellucida. Now, you'll need some more arsenal from your acrosome. Okay? Here, it's a different set of enzymes because the zona pellucida is different. So the different set of enzymes that will cause the lysis of the zona pellucida are the acrosin, esterases, and neuraminidase. These enzymes are released from the acrosome of the sperm and used to forge a path through the zona pellucida. Now, the next stage is the fusion of the cell membranes of the oocytes and the sperm. This fusion causes changes in the zona pellucida itself that prevents other sperm from being able to enter. It's like, all right, one person got through, that's it. The plasma membrane and the mitochondria remain in the fusion site while the tail and the head of the sperm make it inside. I like to think of it as the sperm shedding its scuba gear, if you will, right? The wetsuit and the oxygen tank. The sperm phospholipase C-zeta causes calcium concentration changes that trigger cell cycling in the oocyte, and that kicks off our next stage. The next stage would be the completion of the second meiotic division of the oocyte, and the second meiotic division forms the mature oocyte and a second polar body. The nucleus of this mature oocyte is the female pronucleus, and as it grows, it duplicates its DNA. If we now have a female pronucleus, then obviously the next stage of fertilization will be the formation of the male pronucleus. Within the cytoplasm of the oocyte, the sperm chromatin undergoes decondensation to form the male pronucleus, which also duplicates its DNA, and the tail, well, we don't need it anymore, so it degenerates. The pronuclear membranes fuse, combining the 23 chromosomes from each of the female and male gametes into 46 chromosomes that arrange themselves to begin the cleavage division of the zygote. As the zygote cleaves into daughter cells, first 2, then 4, then 8, the daughter cells are all smaller, and therefore they're not increasing the size of the developing embryo. Remember that zona pellucida that changed to prevent other sperm from entering after fertilization? Well, we could also think of it as creating a sort of cocoon that protects this initial zygote as the cells initially divide. This compacted mass is called the morula when it is between 12 cells and 32 cell stage. It starts transforming into a blastocyte, and the zona pellucida degenerates around day 5, allowing the blastocyte to enlarge considerably thereafter. Now, we can take this process of fertilization out of the body, right? We call it in vitro fertilization, and that involves the collection of the oocytes with an aspirator. And typically, we will do hormone stimulation so that we could have multiple mature oocytes instead of the typical one that would happen with normal ovulation. And these oocytes are then placed on a petri dish with capacitated sperm. In essence, if a couple is struggling with the sperm getting to the egg, then we'll bring the egg to the sperm, if you will. This is an invaluable tool, especially in cases of infertility due to either a blocked uterine tube, but also in cases of oligospermia. And oligo is just a Greek root that means few. So even with a low sperm count, in vitro fertilization only needs a few sperm to be successful. Now, cleavage of the zygote happens in the culture medium until the 8-cell stage is reached, which you can see here. And then these embryos get transferred back into the uterus. The technique of intracytoplasmic sperm injection, or ICSI, involves injecting a sperm directly into the cytoplasm of a mature oocyte. Now, given your understanding of fertilization, what are other things that could cause infertility that might be resolved if you could do an ICSI?